Naomi. This is a real honor to be invited to give this talk this morning. And I'm going to give you 4 pieces of talk that are sort of loosely connected. And where I'm going to start is with what Naomi asked me to talk about, which was a little bit about the history of the software development that came first out of my work and then out of the work of the lab. And that piece of the talk risks embarrassment in a couple of ways. One, I don't want to fall into the trap of talking about nostalgic, good old days stuff when I was a postdoc, but that's what I'm going to talk about. And second, the actual story that I'm going to tell you is mostly the truth, which means that it's a little bit embarrassing. And I thought that the embarrassing part of it would actually be useful to you, though not to me, about how exactly some of this work came about. I think a lot of the people here are just getting started in the field, and I hope that it might be useful to see that one can stumble through a series of accidents and still end up in a decent place. So where this story starts is that I was a PhD student, and I became interested in a particular problem. Anybody who's followed controversies around junk DNA and code will now have some insight to where my point of view is coming from. At the time I started my PhD, this bacteriophage, the wonderful bacteriophage T4, had been found to contain 3 introns. It was absolutely inconceivable that a bacteriophage with a highly streamlined genome would have introns. So these introns had to be doing something useful. Everybody believed it. They had to be a key to understanding where introns came from. Anybody who figured out what wonderful regulatory event they were being used for, this bacteriophage, would be a hero. There were 3 of them, and at the time, we knew almost the complete genome sequence, even back then, of bacteriophage T4, because of work by Betty Cutter and many others in the phage T4 community. 160 kilobase genome with a circular genetic map. 3 genes contained introns, and they are all in nucleotide synthesis, thymidylate synthase, NRDB, in the nucleotide reductase, and what turned out to be the anaerobic nucleotide reductase, a gene called SUN1 at the time. So that little conspiracy theory that they're all in nucleotide synthesis, it turns out that they are introns that require a GTP cofactor, which is a nucleotide, and you could spin all kinds of regulatory stories about what the introns might be doing. We spun those stories. We believed that these would be regulatory elements. And one of the questions was, so let me just say, because I'm going to say one part of the story, the other part of the story is these turn out from work that was not done by me, done by Marlene Belfort's lab in New York, that these are all mobile elements. The introns are actually hiding the fact that the insertion is in place. So once the insertion goes in place, it splices itself out. It's a transposon that's totally silent. What's sitting inside the intron is an open reading frame which catalyzes the mobility of these things. So these are transposons that have evolved the ability to splice themselves out of their target genes. And they're highly site-specific, they're hard to get rid of. In a mixed infection of intron plus and intron minus, everybody comes out intron plus. So the reason they're there is because the phage is unable to get rid of these little molecular parasites. One of the questions, though, was, given that there's 3 of them, how do we know that there's not more of them? It would be super interesting to find more of them. So how hard could that be? It turns out these are not just any intron, these are catalytic introns. They're group 1 introns, and they have a very distinctive RNA secondary structure. So any biologist faced with, say, a structure of an intron, another intron, and say, it's a group 1, it's a group 1, it's a group 1, based on this distinctive folding pattern. But the primary sequence that's shared across the great expanse of all group 1s is very little. There's a conserved G in the catalytic core, and that's about it. So it's the structure that makes a big difference if you're going to look for more of these. And that's what I wanted to do. So I went around, I asked I was an experimental biologist, I played basketball with some computational buddies, Gary Stormo, George Hartzell. George Hartzell in particular, who Naomi probably knows from Berkeley, taught me how to code, what have you, and I started worrying about, come on, how hard could this be? The computational geeks must have figured out a way to take the pattern of conservation of an RNA, which is that the sequence itself might not be conserved, but say this column and this column, if they're Watson-Crick base paired, are in a very particular pattern, that GC would be substituted by CG, would be substituted by AU, what have you. Anything that could look at the pairwise conservation would see information that the Watson-Crick pair was being conserved. So surely there exists a sequence alignment procedure that would take that base pairing pattern and use it as a search query into the phage T4 genome, in my case. That didn't exist. Gary and George were like, no, we've never heard of anybody solving that problem. And I sat down and started re-implementing for myself algorithms from the field, trying to figure out, again, with the hypothesis that, how hard could it be? One thing that I took off from was a simple pattern searching program. So regular expressions are easy to extend a bit into taking into account base pairing. And there was a great program from Danielle Gauthier called RNA-Mott that was being used for search. It was a little slow. And I just took regular expression code from Henry Spencer from a widely used Unix implementation and adapted it to doing the same kind of thing. And that was my entree into doing this. That's not a very powerful way to search. These are not statistical models. They don't allow much. They allow mismatch, but you have to say which mismatches you're going to allow. And it was sort of a pain to manipulate these pattern search programs to do what you wanted to do. And in the end, I couldn't do it. And I had to give up. I didn't see a way to do it. There wasn't any literature on how to do it that I could find. And I put that aside and I went to do a postdoc in something completely different, which I'll briefly mention in a second. But as I was leaving for my postdoc, Gary Stormo gave me the technical report that was coming out of Anders Krogh as a postdoc and David Hausler's work about hidden Markov models. And this sounded pretty cool, but I put it in my bag. I was moving to Cambridge, England. When I got to Cambridge, England, Richard Durbin gave me exactly the same technical report. He said, you got to read this in your copious spare time. And because I got it from two people who I thought were terrific computational biologists, I figured I actually ought to read it. So in my copious spare time, I was reading. And something that I've heard a lot that I actually don't believe in is the dictum to don't reinvent the wheel. My whole career has been about reinventing the wheel. When I started writing hidden Markov model code, I was advised by people in Cambridge, because there's a big Cambridge speech group, to use what's called the HTK, the HMM toolkit that was developed by Cambridge Engineering for speech recognition. It's a widely used, readily adaptable HMM toolkit. And I decided I was going to write it for myself. And I think other people have had the same idea and come out reasonably successful. Santiago Ramon y Cajal, in one of his autobiographical books back in 1916, says what he started doing was he just said, I'm going to dive into a field where people don't fully understand what they're doing, but there's good stuff already. I'm going to try to implement it for myself. In his case, a set of experiments having to do with Golgi staining of neurons, to try to ask the question whether neurons were individual or whether they were all one big network. And he was able to answer that question, and that was pretty fundamental to neuroscience. So Hammer starts then as a side project, as a re-implementation of a published tech report from Anders Krogh. And I had essentially no new ideas other than little ones as I started to do that work. As I started to do that work, I realized that there was a solution to the problem that I was really interested in originally, which was this RNA search problem. This is a copy right out of my notebook at the time I was figuring this out. You could make HMMs work on a tree structure. What I was inventing here, if you know the jargon, is a stochastic context-free grammar, which I didn't know about at the time. You could have states that generated 2 residues at a time. You could allow insertions between states in a binary tree. You could make that thing work. You can see me. I've figured out that it's going to be n-cubed in memory, this algorithm. I'm figuring out how many states it's going to take to model a tRNA. This was like the first couple of days of suddenly realizing one of these sort of nice flashes in one's life. Oh my God, this actually works. There's a dynamic programming algorithm to do this. I showed the speech engineers from Cambridge this, and they said, that's a stochastic context-free grammar. Don't you read the literature? But I liked it, and it was actually going fairly well. And my real project was not going well, in the sense that what I was really trying to do was developmental neuroscience in C. elegans. And I was trying to label individual neurons so that I could follow them growing out in real time in a developing embryo. Anybody who knows the biology and neuroscience now knows this is completely done. It was done by discovering green fluorescent protein, which was published first by Marty Chalfie. I had it in my hands. I was doing GFP experiments. I was doing essentially the experiments that Marty's lab did. I went to a worm meeting one summer, and Marty took me aside and said, we already have this patented. We already have a paper in Press in Science. What are you doing? How did you get the clone? You're not supposed to have a clone. And I thought, well, this is a very bad position for a postdoc to be in. Marty went on to win the Nobel Prize, and I went on to write a progress report to my postdoc granting agency saying, well, I'm in a bad spot. The work that I set out to do for my postdoc has essentially been scooped by the Chalfie lab and by actually several other labs have GFP in their hands at the time. But I had this side project going that seems to be okay, and there was a sort of moment when I had to choose to be a computational biologist and switch, go all in to do computation. Now, the thing I want to say about this is that I had essentially zero confidence. I expected my grant to be cool. And my postdoc agency, which was a neuroscience granting agency, the HFSPO in Japan, did not even blink. They continued to support me as a postdoc. And so, without going into a lot of it, there's work that came out of that postdoc in the early years of my faculty career at Washington University that, I'm pretty proud of. The Hammer came out of it, the Stochastic Contextfree Grammars came out of it, and a book that I wrote with my friends Anders Krogh, Graham Mitchison, and Richard Durbin came out of it shortly after I started my faculty career. The point I want to make from this nostalgia is that, okay, several things. This is sort of a half-assed way to get into computational biology. I'm not a computer scientist or a mathematician, I'm a biologist. My biology project was destroyed. I got into this as a side hobby, and I was doing something that biologists will tell you not to do, which is develop methods and tools instead of fundamental science, having had my fundamental science project completely destroyed. And all I had really done, though I thought I invented SCFGs myself, of course that was widely known in the speech engineering community, and so I really hadn't done anything that wasn't already in the literature other than details. So I will now argue that, because it's the only thing that I know, that these are ideal conditions for making an impact as computational biologists. And I emphasize that the personal history colors one's way of looking at what's right. And so I wanted to give you this sort of embarrassing personal history, partly to say I'm in no place to give you advice. But I do think that, at least for what's happened to me, that these conditions set up a lot of the way I think. And so you can have this in your head as I sort of give you things that you can take at least as my ideas, if not advice. So, again, biologists will tell you, don't reinvent the wheel, don't develop tools, don't make sure that you're making a huge step forward, don't do incremental science. And the thing that makes me the most crazy about those things is that don't do incremental science. There's a lot of things in the world that move forward as a combination of leaps and incremental improvement. And especially when you're talking about engineering, when you think about what's happened with something like 100 years of engineering of cars, there are fundamental advances that have been made in cars, but a lot of the sort of motion from the Model T to a modern car is incremental, is the very soul, heart and soul of incremental engineering. And when you look at things, like in the field of sequence analysis, and I would argue look at anything, look at Moore's law or any other exponential curve of technology and engineering technology getting better. You can start with sequence analysis in the 1980s with how Russ Doolittle knew how to do it from a science paper that was particularly influential in like the mid-80s, where people were using a +1, -1 scheme with basic dynamic programming. And if you benchmark this is just a particular benchmark, percent identity of pairs out, so at what percent identity can you detect 50% of a benchmark? And I won't go into the details of the benchmark. As we developed things like proper score matrices, Blossom 62, BLAST, which actually is better than Smith-Waterman, counter to popular writing, because of a lot of stuff, including bias composition corrections that are in BLAST compared to most implementations of Smith-Waterman, PSI-BLAST, and then finally profile hidden Markov models with probabilistic scoring schemes. This does have some technological leaps in it, but it also has a lot of incremental engineering. If you sat down and you wrote a profile HMM code like I wrote in 1993, that would have all the fundamental ideas of profile HMMs. But when we compare what Hammer 3 does to what Hammer 1 did 20 years ago, it's night and day. Hammer 1 would be somewhere over here. There's a lot of other engineering that goes into this code to actually make it useful. And so that means that, so the point I want to make is that there's a big difference between science and engineering. And the engineering is valuable. Science, I'm not dissing science. There's science in computational biology. There's fundamental algorithms, fundamental mathematics that comes in leaps. But there's also an awful lot of engineering. And I think the engineering gets short shrift because of all this biologist, oh, don't do incremental stuff. The result of the science is, of course, a paper. The result of the engineering is a piece of software, or in my case, a piece of software. And there's a lot of stuff that has to go into that software to have it have impact. It has to be usable. It has to have a long life. So Hammer and Infernal have now been around for 20 years, God help us. They need to be something that you can integrate in other stuff. So they have to have a relatively simple interface that people can build on. And they actually have to solve a problem pretty thoroughly. There's a lot of things that we do that are sort of a halfway solution. And you end up with like 20 applications that are doing the same thing, and nobody's happy with them. And then someone comes in like the XKCD and Cartoon, and now we have 21 solutions to And so until somebody goes all in and says, I'm going to devote a significant chunk of my life to this is an important problem, and I'm going to be the one go-to application that solves this thoroughly, that's when something actually moves forward. And the steps that it takes, the level of commitment that it takes to solve the problem thoroughly is a slightly insane level of commitment. You really have to say, I'm going to go all in on this because I think I can be the one application. Or in Profile HMS, for instance, there's like 2 or 3 implementations. And so what that leaves me thinking is that there's a fallacy, there's a big fallacy in the community, which is that you're rewarded for how many publications you have. How big is your CV, your tenure review committee, blah, blah, blah. The truth, at least the truth from my perspective, I would hesitate to say, go to your tenure committee and tell them that Sean Eddy said it. But at least in my case, I made a series of bets that paid off. I bet that this was true. I bet that you are rewarded in science, in our field, for how much impact you have on the community. And so the consequence of that is that it actually is worth the time and effort to identify a place where you can make an impact and go all in and say, by God, I'm going to solve that problem thoroughly. So I deliberately think about this as arbitrage. I believe, if you know this, so it's the practice of taking advantage of a value difference between 2 markets who have incomplete information. Academics believe that their career system only values publications, but in fact, the research community does demand strong computational tools, which means somebody can play the middle. Somebody can say there is a vast literature of advances that are being made where individual ideas are in the literature, but they're not in a form that have been integrated in tools that the researchers can use. I'm coming from the perspective of being an experimental biologist. I like to take those ideas and put them in a form that my colleagues in biology can use, and that's been a productive middle ground to inhabit. So, for example, when we got to Genelia Farm, one of the projects that I decided to take on and sort of place my bets on was something that was not, so Genelia Farm's a neuroscience place. It's a Howard Hughes Medical Institute. It's all about fundamental science and revolutions and non-incremental science. And we deliberately took on an engineering project, which was to say the single thing that I know that we could do, or at least it's worth betting that we could do, that would make a big impact on the community. Profile HMMs are slow, they're 100 times slower than BLAST, and if somebody could figure out how to make them fast as BLAST, that would be the last reason why anybody's using BLAST instead of all the great theory that was developed by Krogh and Hausler. So let's figure out how to engineer a piece of software that would go super fast. And I don't know how to do that, I'm not a computer engineer, but other people in the community do know how to do that, so I read the literature. And one guy in particular, Michael Farrar, who eventually joined our lab, was working for a telecom, and in his copious spare time, he was developing vectorized implementations of Smith-Waterman. This particular paper caught my eye, and it caught Bill Pearson's eye. If you haven't noticed, the Smith-Waterman implementation in FastA these days from Bill is as fast as BLAST for the same reasons that Hammer is now as fast as BLAST. Both Bill's lab, both Bill personally and me, used code from Michael Farrar, and so we take advantage of modern chips and vectorization and what have you. I learned the basic techniques of this from reading Michael's paper, and that was sufficient to then spend a lot of time engineering it in Hammer. And I recently went through a use review and had to stand up in front of a bunch of people and explain why I did this, and again escaped the axe. So I still believe that this is the right way to do things, though your matters may vary. So another thing sort of loosely connected to that is that because of this philosophy that we're in it for the long term, we're not doing a quick strike, writing a paper, another quick strike, writing a paper, we've had to make engineering choices in the projects we've taken on that maximize the probability that we're going to be able to live for the long term. So one of those, for instance, in the way that we did the PFAM database, is a very simple idea that had a big impact on how we deal with the exponential growth of the sequence database. And it's an idea that I think ought to generalize to other things. So the problem was that lots of people were clustering the database and making protein families. Then the database would grow for a year. And the PI would go back to the student or postdoc and say, okay, now we got to recluster the database. But because the clustering was never perfect, there was always a lot of curation, a lot of work. It's like, I'm not going back and doing that project again. And so there was a lot of clustering. Protein family database would come up, and then they'd go back down as they went out of date with respect to the database. So once we had things that were good probabilistic profile methods, Hammer, Inferno for RNAs, we could adopt a strategy where we had these little curated seed alignments, these little representative alignments, like 20 or 30 sequences, sufficiently representative that you could build a profile of them and then sweep the database, however big it was, and use automated methods to find what PFAM and RFAM call the full sequence alignment. So a very simple strategy that sort of separates the curation from the growing database by taking advantage of tools that are sufficiently scalable they can sweep the whole database. Now, even that curate, so now we can curate things that are relatively stable objects. So we can curate a database as big as 15,000 protein families. We still don't do a particularly good job of that in the PFAM database, and so now we're trying to find other scalable ways, and there's a collaboration actively with the Wikimedia Foundation to do this inside of Wikipedia. PFAM and RFAM were some of the first biological databases to directly use Wikipedia. All right, a third part of the story. And so the loose connection here is because the lab, has taken a strategy of non-traditional impact, at least as far as biologists are concerned. So when I get up and defend my lab, I'm usually defending it saying, we have Hammer, we have Inferno, we have PFAM, we have RFAM. There are a set of resources that are the core of our lab. We also have biology, we have papers, so I'm sort of backed up, we have a portfolio. But those are the main things that I talk about from our lab. And this has made me think a lot about scientific publication and what we're trying to do with scientific publication, and it's led to me serving on whatever committees for the future of scientific publication. And so I want to say a few things about this that I don't think are widely appreciated. Maybe they are, and I just don't know it. So the first scientific journals show up in 1665, two of them actually. This one, which I can't pronounce, it's French, Journal des Savants or whatever, was set up because people no longer, there was too much information in the world. And so somebody needed to summarize it. So this was essentially a review journal. It became more of a Scientific American, and then it became more of an Omni, and then it disappeared. This one stuck around. The Philosophical Transactions of the Royal Society of London, founded in 1665 as well. So the idea was in the air that the world needed journals. Sir Henry Oldenburg was the editor, and what Sir Henry had been doing up till that point was sending letters back and forth. So he was a hub in the scientific community in Europe. People would tell him what they were doing, and then he would send letters to his buddies, say, guess what Newton's doing? And so that was the mode of communication. It was all flowing through Oldenburg and a few other hubs by letters. Oldenburg decided this was a crazy, non-scalable system, so he decided to publish a journal, and he instituted peer review. It all comes from here. There's a great, you can go look it up on the web, a great letter. He convinces Newton to submit some of his optics work to the journal. Newton has never had peer review in his life. Gets his reviews back. They ask for a bunch of stupid stuff. Newton writes a letter to the editor saying, I'm never submitting a journal again. Totally stupid. What are these reviewers know? They're idiots. But it set up something that I think is it's the heart of the system. So there's two parts of the system. One is peer review, but the other is the quid pro quo. There's a moment in time where your paper gets accepted, there's a quality control check, for better or worse, from peer review, and then it's like you are giving the scientific community something, and then the scientific community is giving you priority. And this is how he convinced Newton to do it. He said, look, you're doing all this, because if you don't know this, they were doing all this crazy cryptography. People were writing their theories down in code and depositing them in sealed letters with their lawyers. and that this was how they were assigning priority, that someone else would, Hook would say, I discovered something about optics, and Newton would go, ha ha ha, and call his lawyer, and then would produce the sealed envelope and say, actually, well, I discovered that 2 years before you. And that was not a very productive way of getting information out the door. And so what Oldenburg did was invent this system saying, okay, we'll publish your paper. That establishes the marker that says you did it first, but in exchange, you've got to actually tell us what you did instead of writing it down in code on a gold ring. That system still exists. That quid pro quo is, regardless of however cynical we sometimes are, is widely accepted in the scientific community. I served on a U.S. National Academy of Sciences committee in 2003 where a bunch of people, including industry representatives, agreed on this, that the fundamental purpose of publishing scientific information is to move science forward. In particular, that there's the idea that there's a key result that you're trying to deliver. You're basically advertising, this is what I've done, this is what I think is going to be useful. Your reviewers say, yes, we agree that's useful, and we agree that the author is delivering it in a form that we can understand it, verify it, replicate it, and build on it. That is, you can actually use the information. It's not, ha ha ha, I did this and now you can't do it. It's, this is something you can build on. Now the problem we were tasked to address is what happens when the sequence is no longer in figure 1, but the sequence is 3 gigabases of the Celera human genome, and so when science publishes the Celera human genome, is science obligated to make the sequence available in GenBank? And the committee came down resoundingly, of course it is, because that's the purpose of scientific publication. So my argument here is that as we talk about open science, which is a very important thing to be talking about, I want to talk about it in the context of, this is not new. This is the intent of the system. This has been the intent of the system since 1665. The reason that we're so exercised about open is because it doesn't fit in figure 1 anymore, if it ever did. There's a lot of things, like large data sets, like the software, like materials from biologists like transgenic mice, and the literature itself, where the result is not something that is just distributed in a PDF, but actually requires electronic distribution. And so the battle that I've been fighting, that other people have been fighting, that I'd like everybody to fight, is to convince the journals, and they aren't convinced and they're just worried about their money and blah, 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 that they have to not only deliver the papers in PDF, but to deliver the fundamental results of the papers in a way that can be verified and built upon, which means we need the electronic files, blah, blah, blah. And this is one of my favorite quotes, sort of summing all this up. This is from David Donahoe. I think a lot of people have seen this because Robert Gentleman has picked up on it too. In computational science in particular, though not limited, I mean, transgenic minds, the paper is an advertisement of the scholarship. It doesn't deliver the result. It says there's a result here, go and get the software, go and get the data set. And so that's a different way of, slightly different way of thinking about the publication process that we and our journals have to deal with. All right, finally, what I'll call 2 magic tricks. And this is totally spurious. I come at this as an experimental biologist. There are things that I still do as a biologist that I carry over from my biology training. I don't think that these are super sophisticated. But I'm sort of shocked by how much people are shocked when we do. And find interesting pieces of stuff. So let me just start here. What we're doing at Genelia Farm in part of my lab is we're actually doing neuroscience again. So we're doing experimental biology. And I just want to say, if you can see it, it might be a little dim. So GFP, which I got scooped on, now works like gangbusters. We have it in multiple colors. We have it hooked up to GAL4 UAS lines and Drosophila in this case. And people like Tzu-Min Lee at Genelia Farm can use GFP to label individual neural cell types. And this thing is the lineage of one neuroblast, so one neural stem cell, going through 42 divisions, where at each division it determines another cell fate. And this is remarkable. I mean, everybody knows development's pretty remarkable to begin with. How does one genome make all the different cells of the human? But here's one cell, one lineage, where it makes cell type 1, cell type 2, cell type 3. If you pull it out of a dish and you put it in vitro, it'll recapitulate those divisions. It's running an internal program. programmed by its transcription factors or whatever, we don't understand that. That's a sort of another sort of seriously kind of thing, like the group on introns and T4. It's like, I would really like to understand that. Now, in trying to understand that, that's put us in a mode where we're sort of like, so now I'm back to being a biologist. What we're doing, Lee Henry, Fred Davis, and to some extent me, though mostly these guys at the lab, we have a nice technology, also developed by Steve Henikoff's lab, for labeling the nuclei of cells with epitope tags. Nuclei are super stable, little Death Stars. You can isolate them out of ground-up flies, and we can then do a magnetic bead pull down on these nuclei. So now we have 20 specific nuclei out of the fly, and we can do an encode project on a particular nucleus, so on a particular cell type. So instead of just like fly heads or stage 3 larvae or something like that, we can now say these particular neurons, what are they expressing at the RNA-seq level from the nuclear RNA, ChIP-seq, what have you. And that puts us back in the game of how do we analyze the data we've got. So we're using great tools that are produced by people in the community for mapping reads, for calculating, how what the RPKMs are, and so we're now a user of tools as we do this stuff. And a thought that's been going through my head as we do this is that there's a big difference between, so a lot of what we talk about in computational biology is sophisticated infrastructure. Taxonomies, ontologies, software engineering, building pipelines, and that to me is like building interstates. You want to get from point A to point B, you want to get there super fast with a big load, you need to be a really good engineer. But a lot of biology is sort of sitting in, I'm taking a walk through the woods and I'm looking at random stuff and I'm trying to figure out what's interesting. And for that kind of stuff, the big pipelines actually get in your way. So what we're doing, so like I'm on the advisory board for Galaxy, and James Taylor and Anton Nekritenko are like, are you using Galaxy to do your RNA-seq? I'm like, are you kidding? Because we want to be close to the data. We're using the command line. We want to actually see what's going on. Maybe once we get into production mode, we're going to use Galaxy or what have you. But in the meantime, we're using really robust tools as our read mappers and stuff, but then we're actually using Perl and Python scripts to glue it all together. And I'm entirely unashamed by that. So the two magic tricks that come from biology. And I don't think, again, I'm not trying to say that people don't know how to do this, but I'm sort of surprised. I think that this is underused. So you see, so one, there's a lot of things I can sort of go off on, but let me go off on two. One is people will give statistical tests, they'll do their computational analysis and they'll say, oh, p-value of blah. And these p-value things, these statistical tests, they were developed by Fisher, what have you, back in the 1910s when we didn't have computers, and they were developed because people didn't know how to crunch the numbers big enough. These days, a Python script can rip through and do a simulation and directly, like, here's a simulation of what the null hypothesis is. It forces you to write down what your null hypothesis actually is. And then you just count how many false positives you get in the simulation of your null hypothesis. And you can burn computer time to do something that requires, as opposed to getting the statistical test right. And it's surprising how often this would have saved people. So we do both. I mean, obviously we use statistical tests all the time, but we do both. It's easy to design control experiments to test things, like this particular statement out of a very important paper, the early sequencing of the yeast genome. They chose this famous cutoff of 100 amino acids. So why do we use 100 amino acids as a cutoff? For anything above 100 amino acids is coding, anything less is non-coding. A lot of the literature still uses this stupid number. It assigns lots of the ribosomal proteins as non-coding RNAs. It's a totally idiotic number. The reason that it comes is because of a calculation that was done by the yeast group early in the day, saying that is really rare. It has less than a 0.2% probability of occurring by chance in Cerevisia. That number is obviously wrong. Shuffle the yeast genome, translate it, save all the ORFs longer than 100 amino acids, and count them. There's 383 ORFs longer than 100 in your simulation. Not coincidentally, a few years after the yeast paper, there were other papers written saying, boy, there's like 400 open reading frames that seem to be different. We don't see them expressed, we don't see mutants in them, and it's because they were false positives, because this calculation was messed up. How was it messed up? It's the calculation per starting point that you would have in a 100 amino acid open reading frame, and now you have to multiply it by the size of the yeast genome, which happens to be 14 bytes. So there's some sort of obvious things that one could say about this. And again, they fall in the category of I am totally unashamed to reinvent shuffling algorithms, to reinvent translation algorithms. we as computational biologists, I think, should feel unashamed about building up a repertoire of our toothbrushes that we do not share with each other, because they are things that we know and trust and know inside out. We've actually done controls to know that they work, which you can't even always rely on. There are, again, I could name a famous paper that the translation program had a major bug in it that led them to miss obvious open reading frames and call a bunch of non-coding RNAs. The ability to do these kind of analyses, of course, presumes that I can get these sequences on my disks in the first place, and so I have to be able to go to the used paper and get that. So our ability to build on results relies on the data, that's obvious. And I am also unashamed to be using the command line for the majority of my exploratory, non-production scale work. Second magic trick, if you will, that I think is underused, and it's the main thing that I advocate these days because I think it would save a lot of people from traps in their large-scale analysis. But again, a fairly obvious idea is to downsample the data. So you can have however big data set you want, take 10 samples out of it and actually look at them or get a biologist to look at them. And the number of times that this turns up an error, because you look at it, you go, oh my God, 5 out of the 10 are an obvious artifact, and I got to go back and redo that pipeline. The number of times that happens is large for us, and we go back and reiterate our analyses. I've had one famous ENCODE biologist tell me that this was a completely invalid procedure to test ENCODE data with, because even if 10 of them were bad, what about the other 9,999,000? It's like, what part of random sampling? And let me give you just a specific example of it, and so why I call this a magic trick, because this is a thing that I caught in review. I was doing the peer review, and actually a couple of the reviewers and the editor responded in total incredulity, if that's how you pronounce it, saying, how the hell did you catch this? Because it actually required looking at the data that these guys were trying to publish, which reviewers apparently don't do. And so the paper was about microexons, and what they had was they had a alignment procedure, a spliced alignment procedure. It was going into Drosophila and aligning RNAs back to the genome. And there were a lot of them. They predicted a lot of microexons. And actually, it was a pretty great spliced alignment program, and it worked on controls, on their positive controls. It was obvious that it worked. But as you pulled data out of it, it was obvious it was missplicing, because the splice consensus is GTAG, and these were not GT. This was GT. That's not really. So there's something that wasn't quite right about this. And what I noticed was, as I looked at, because of a small number of them, I could look at them, that they were misaligned. And the reason they were misaligned was because the splice aligner was assuming that the RNA came from the reference Drosophila sequence, and so it shouldn't have any changes. So it was tuned to align with 100% identity. So it had to force this. There was a SNP. So it had to force the alignment to avoid the SNP. And when it did, it created 2 microexons in a tiny little intron. But in fact, that was a canonical intron sitting there. And it was just a result of, they didn't expect, and actually I'm still amazed by the diversity in Drosophila strains. There's enormous heterogeneity in Drosophila compared to the Berkeley reference. That's just the way it is. I didn't expect it, they didn't expect it, but it was easy to see once you subsampled the data. So sort of in closing, Lincoln Stein gives terrific talks about the future and past of bioinformatics. And one thing that Lincoln has said back in the day was that bioinformatics would be gone by now. And I sort of agree and disagree. So that is, I think there's a horrible history that we want to avoid of not outsourcing. Biology is tempted to outsource its data analysis. They've done it to biostatistics for clinical trials. But what they didn't do was they didn't outsource molecular cloning. There was a moment in time when the molecular cloning guys were super popular. Everybody needed a molecular cloner in their department to be able to do cloning, because nobody wanted to learn to do it themselves. But eventually everybody learned to do it, and it mainstreamed. I think a lot of sequence analysis is mainstreaming now. We're seeing people growing up, you guys, biologists, computer scientists, everybody's learning how to do the science. And the data analysis needs to be part of the science. So I don't think of this as interdisciplinary. I think it is part of the discipline. To do the science, we have to do computational analysis. It's just the business of biology. So biologists, anybody who's doing this, whether you've started in computer science or not, you're a biologist if you're analyzing data. But at the same time, strong computational tools are always going to be in demand. So there is going to be a field. I think Lincoln also made this point. There's always going to be a field for people to develop the sort of strong tools that we can harness to do that analysis. And with that, I will stop, and I am working with a terrific little laboratory at Camellia Farm. There's also a Hammer Web Services team at Camellia Farm led by Rob Finn, and a lot of what we do is supported by a collaboration of people, especially at Cambridge UK, but also elsewhere, largely run now by Alex Bateman for the PFAM and RFAM databases. And I'll stop there, and I'd be happy to take questions.
Thank you for the talk. I was wondering if you could comment on some issues that I think will be of interest to us as open source developers regarding trademarks and defending your brand, because you've taken quite a strong stance on that with Hammer. Yeah,
so, well, I don't know. I hope it doesn't come across as too strong. The business of dealing with industry has always been fraught. So you want your methods to be used, and you want them to be, maybe I should say, I want the methods to be used. I want industry to pick them up and use them. But at the same time, there's a sort of mode of operation that the industry can get into where they're trying to leverage you a little too much in a way that I think is unfair. So where I decided to draw the line, for example, was at the name Hammer. It's like if you're calling your thing Hammer, it ought to be Hammer. It ought to be up to date, it ought to be our code, it ought to be a single code base. And if someone has like re-implemented this stuff, and it's their code, and then they attach our name to it, it just sort of ticks me off, actually. So that's why we got a little militant about the trademarking and stuff like that. We figured that the code was, had sort of risen to the point where it had been re-implemented by a bunch of industry people who I totally encouraged to do that, but they were using the Hammer name on it and sort of advertising it, what was essentially not even forks, but complete re-implementations of the code that I had to look at, proprietary code, what have you. So I decided it was appropriate for an open source project to try to defend the brand, to make sure that when people are using something, that they actually are using the open source version of it rather than a re-implementation of it. And I thought I could convince most of industry that that was a reasonable position to take, that they could say this was inspired by Hammer, that they collaborate with Hammer, or whatever they wanted to say, but not to represent the stuff as being Hammer. And that's largely worked with the exception of one or two companies.
You mentioned that we shouldn't share each other's, well, perhaps it's not a good idea to share each other's toothbrushes. It's a nice analogy. But who found the bug in the translate tool you were using?
Actually, it was a group at Regan that was using it. I found the bug and reported it to Regan.
So although other people were using it, you were still the only person who had spotted it.
Yeah. I mean, it's obviously a tricky business. I mean, because, of course, it was Webb Miller who used that analogy first that I saw about it's really funny that people treat software, everybody's code like it's their toothbrushes and won't reuse. We all evangelize about code reuse, yet how often do you actually reuse code? I've sort of I don't know what sort of there's some sort of Zen acceptance of this. There is code that I do reuse, and that sort of gets back to code that's complicated enough and outside my space. It's like, I want a regular expression package, I'm going to pick one up and use it, But something that's actually in my space, like, I totally want to understand this, then this sort of equation shifts. And so I think it's sort of there's some sort of spectrum there. And I'm always making a decision of how much time do I want to invest in this versus am I going to be using this for the next 30 years of my career, and it's totally in my space, and I'm probably going to want to tweak it, and I want to totally understand it. And so there's a bunch of little basic tools. And then there's an interesting thing of there's a mode of code reuse that I think we all use, which is you still want to see the other guy's code. You don't want to use it, you want to see it. Because you're going to get ideas like, how do I implement this? So for instance, we use the GNU Scientific Library a lot. We don't want to suck the whole thing in, so we always recode. But we're always looking at it as a reference work to say, how do you implement this special function and stuff like that? And so it's not black and white, but there's a lot of stuff I feel unashamed about, of like building up our own suite of tools.
saying there is that there is utility code, like systems code, and that depends where you are in the stack, what that is. And then there's the innovation code, which is actually really your research, it's your science, and therefore you have to really have your hands upon it. And that kind of comes really strongly. I'm a computer scientist, and that all comes from computer science kind of field. That's what we do as computer scientists. We're engineering. Computer science and engineering mathematics is not science in the sense of discovery. You don't discover anything, you invent it all. So everything has to be a series of stacked incremental engineering activities. So this discipline comes from that world, I think. And maybe that hasn't infected computational biology as much as biology has infected computational biology from the publishing view. Is that what you're saying?
No, I think you put it well. In my experience, as it comes from biology, I couldn't really speak to how much computer science we need to adopt. But that sounds pretty
well,
good.
In computer science publishing, we can. It's great if you reinvent. I have an idea. I write a paper. James Taylor has another idea. He writes a paper. We integrate. We write a paper. We compare. We write a paper. We find something else. We integrate. We write a paper. It's kind of the whole thing is based upon building and comparing. Not about I got there first.
Right. Something else that I think about a lot that I think is related to this. I've just been through this with bed tools, so I've adopted using bed tools from Aaron Quinlan and his coworkers. And to me, that's an example of going all in and solving a problem thoroughly. Because I had my own random Perl and Python scripts for manipulating tabular data and throwing it around and sorting it. And they were all sort of half-assed and worked most of the time, and it was fine, but I wouldn't want to distribute them. And then bed tools comes in, for example, and it's like, this is a totally thorough solution to this problem. I'm not going to have to do this again. So now I'm progressively throwing my stuff away. And that's part of this equation of someone has to sort of go that step of insanity beyond reasonable. I mean, bed tools is pretty crazy. It's a pretty thorough solution to this problem of manipulating text files. And you're like, God, fine, I'll use it. But once something reaches that point.
So you notice that, for example, very frequently that you might have error or snips that might introduce error. Or did you notice now for PFAM and CT or PFAM or even HML the incoming sequences of that sometimes are badly predicted or not very good quality? Did you notice their impact on the quality of PFAM or HML or on your tool?
Absolutely. I could give you a long talk on the error modes, the main error modes with a long tail in these databases. And so any kind of sequence database of that sort is a very unsatisfying business for a perfectionist because you see the errors in it and you have to accept them just to get the job done. And so actually one of the main error modes right now, the one that disturbs me the most is not in PFAM, but actually in RFAM, the RNA database. Models are going into RFAM where, although the database is supposed to be curated, the number of structures that we really know for RNAs is so small that a very tiny fraction of RFAM is actually reliable, manually annotated RNA structures now. And so what most of the RFAM structures are actually predicted structures using common tools in the community. RFAM has become adopted by the community as a benchmarking tool for evaluating the accuracy of structures. I don't think people will fully take it on that their structures are largely generated by the same tools that are being benchmarked. And so that circularity is driving me crazy right now. But as you say, pull in almost any PFAM alignment and there'll be at least a sequence often that you look at and go, how did that get in there?
You don't have problem with the limits, the limits of the PFAM with this type of error.
And this is a problem with Hammer 3, for example, the switch from Hammer 2 to Hammer 3. If you're sort of inside this, you know that Hammer 2 did global alignment and did a lot to get the edges of domains right. Hammer 3, because of the speed, we had to sacrifice that temporarily, where temporary means years. And so now we're working on bringing back that global alignment to try to get that accuracy of domain definition back. That ended up being another big engineering project that we're still in the middle of.
how much over time, how much outside contribution was there, whether desired and he didn't accept it, or that actually got into Hammer as a code base?
So in terms of actual code, there's relatively little code in Hammer that's not mine, or there's a little bit from like people, especially these days, more is coming from people in the lab. But there's been an awful lot of input, where actually surprising input from people, say, I don't know, Intel and Microsoft. I mean, because Hammer now is part of the spec benchmark for chips. And so once you're in the spec benchmark, all the compiler guys are upset that you're making your compiler look bad, and all the chip guys are mad that you're making their chip look bad. So then there'll be engineers from Intel and Microsoft and IBM assigned to look at why they're looking bad on the spec benchmark. And then those guys will write to me and say, did you know you could rearrange this loop and you'll get more speed? Did you know if you tweak this flag, and so I get a lot of that kind of email. And it's still my job to write the code in response to that. So it's not a patch, it's not it's not a pull request or something like that. But it's a tremendous amount of highly professional input that I've gotten. And I sort of give open source credit for that. Usually I won't hear from these guys until they've already had a team of engineers studying the code, because they can just download it and look at it.
How do you test the correctness of the implementation?
I don't believe in correctness. The number of times that we find an error, so we have unit testing, we have integration testing, we have nightly testing. The whole thing gets tested on multiple platforms in a scripted nightly thing. There's about as much test code in Hammer as there is actual code in Hammer. And as far as I'm concerned, that just means there's twice as many bugs. And so we need test code for the test code, and it's an infinite regress. And so, because half the time, I mean, seriously, half the time that we find a bug with the unit test, the bug is actually in the unit test code and not in Hammer. That's similarly the case for bug reports that we get from the community. 90% of them are bugs in their stuff rather than our stuff that they still have to all be tracked down. So it's always a mess. So the answer is that we do a pragmatic, we have a lot of code coverage, we measure our code coverage, we have unit tests, but the number of times that I've been surprised by finding a bug that our tests missed is very large. The number of code paths is still large, even though it's a relatively well-defined thing. The number of code paths is a big combinatorial equation. I don't think we can test them all, so we do the best we can. But that said, we put an awful lot of, there's a lot of test code in there. It is a major effort, a major engineering effort in the lab. We take it super seriously, but I'm still pretty sanguine, not confident that it actually works all the time.
Do you have any advice about sort of, I don't know, or just a story about the transition from the first time you wrote Hammer to when it became widely used? Like your story kind of skips over, like, I was writing this in the red lab, and then bam, now everyone's using it. Sort of how did you manage that transition, or think about that? I guess that's probably a common sort of, people are writing tools, they want them to be used, and how do they get to things where it's widely accepted by the community?
I don't actually know how that happened. I put it up on a LMB, Medical Research Council FTP site, and back in the days of Usenet bulletin boards, I announced it on Usenet Biology, Bionet something. But I think one of the main factors was that I had good mentors around me at LMB, sort of mentors. Cyrus Chothia, a structural biologist, was studying immunoglobulin domains, their structure and how they evolved and what have you. And Cyrus saw what I was doing from the little postdoc talks and what have you, and he just became a tremendous advocate and started feeding me, like, it's getting this alignment wrong, or it missed this one, it should really be this one. And I think actually Cyrus was out in the community talking it up. And I think that's the kind of, if I were to point to one thing, it was that. It was having someone in the institution see that it was going to be useful for his work, and then Cyrus was a tremendous advocate outside the LB for getting it out. So it's a combination of things. I didn't really do anything. It just sort of happened.
Have you thought about trying to maintain your code on, let's say, GitHub or Bitbucket, so that people's contributions are more formalized with pull requests?
So this is a source of embarrassment to me. The answer is yes. I think about it all the time, and I think that I'm becoming an old fart because I don't use these things. And I think of excuses that we have built a system that's now subversion based off of our own, internal repository. Back when that was the cool thing to do, and now I just don't want to change because changing is a big effort. I've got other fish to fry right now, so it's somewhat laziness, it's somewhat inertia. It's also, though, somewhat, I'm not, sort of back to Homer's question, the number of times that it's a sort of chicken and egg problem. The number of times that somebody said, I want to work on your code base, could you make it accessible to me? So for instance, we have anonymous, somebody can just get the code by anonymous subversion checkout. The number of times that somebody's come back and said, I'm really getting jammed here, I'd like to write directly to your repository is zero. But of course, that's a chicken and egg. Maybe if it's sitting at GitHub, people wouldn't see that barrier and they would just start doing it. It would happen naturally. So I don't know what to make of it. Like I said, it's sort of an embarrassing question. I feel like I should be more modern. I should be doing that, but I'm not doing it. My excuse is that I'm not actually convinced that there's a lot of people who want to write to the code base. It also might be a sort of personality thing. I tend to be a perfectionist to the level that I think I might not be an easy person to work with. For people, I'm afraid of that. But I guess another answer to the question is if I'm totally wrong, if there's a bunch of people sitting in the audience saying, geez, I didn't know I could contribute to Hammer, I'd love to do that. If only the dummy would put it on GitHub, I'd start doing it tomorrow. I'd like to hear that kind of feedback.
Okay, thanks again, Sean, for